Okay, so now we get to the meat of the information. This video is going to go into mitosis, the steps that are involved and what happens, and hopefully some tricks on how you can remember them. So first, I wanted to just point out um, the difference between sexual reproduction versus cell division. And when I say cell division, this is what we're talking about with mitosis. So sexual reproduction is you have a mom and you have a dad and they have a child. So the end result is that now you had two people and now you have three people. You still have the original mom, the original dad, and now you have a new person, the child, and that is sexual reproduction. So mitosis, cell division, is a completely different way of reproducing because what happens is this cell itself splits into two and you end up with two identical cells. And here's the nucleus and here's the chromosomes. So you get the idea. So you don't have the original cell anymore. The original cell is gone, and instead, this original cell had split into two, and now there are two new cells called daughter cells in place of the first original cell. Okay, so that's the difference between sexual reproduction and cell division. Now, for mitosis, it is going to be very helpful if you memorize PMAT, P-M-A-T, because that stands for the cycles within mitosis. There's prophase, metaphase, anaphase, and telophase or telophase. And for prophase, if you think of the fact that pro means before, then prophase is going to be the phase when the cell's getting ready to start doing its thing to divide. And then metaphase has an M, starts with an M, so if you think middle, that's when all of the chromosomes line up in the middle of the cell. For anaphase, anaphase starts with A, so think away. That's when the chromosome pairs get pulled apart, each to opposite ends of the cell. And for telophase, there's no really good abbreviation for T that I could think of except for the nucleus, because that's when your nuclear membrane forms around the two new sets of chromosomes. And you'll see what I'm talking about as we go through this. So prophase, this is step number one. Prophase, again, pro meaning before, so the cell is preparing for division. This is before it divides. Now you start right here. This is the cell that you're going to start with. This is your original cell, and um, this is the cell wall right here. And inside this black line, this is the nuclear membrane, the nuclear envelope, which is like the wall around the nucleus. And then within the nucleus are two sets of chromosomes in chromatin form, which we went over in the previous video. And then up here, these little Mickey Mouse ears looking things, these are called centrosomes. Cent, like a penny, centrosomes. And what they're going to do, they start up next to each other on one side of the nucleus, and as prophase goes along, they're going to move apart from each other to opposite ends of the cell until one is over here and one is over here. Now at the end of prophase, we're going to end up with a cell that looks like this. As you can see, the centrosomes have moved to opposite ends of the cell, and the centrosomes grow these little fingers, they're called microtubules, out of the centrosomes going toward the middle of the cell. And if you notice, up here at the start cell, there's that nuclear membrane I was talking about, that black circle, that's the wall around the nucleus. Well, that's not there anymore. The nuclear envelope has broken down and dissolved, and so it kind of spills the chromosomes out all throughout the cell. And also you want to notice that these chromosomes are not in chromatin form anymore. They're in chromosome form. They have tightened up and wound up into short, thick segments, and they thicken up also. So if you look at a cell under a microscope, when it's in this phase, once the chromatin has turned into chromosomes, you can actually see this under a really strong microscope. So this is just more information on what I just went over. The centrosomes, they move to the opposite poles of the cell and begin growing fingers. I like to think of them as fingers because if you see it under a microscope, it looks like interlacing fingers together, but they're called microtubules. The nuclear envelope, which is this black circle right here, it's the walls of the nucleus that breaks down and dissolves into nothing, so it goes away. And then the chromatin, this red and green chromatin up here, the sister chromatids, they wind up into short, thick pairs of chromosomes, and each pair are stuck together at the centromere. So you have one chromosome and its pair, its chromosomal pair right here, and you see how they're stuck together. Let's get a different color. You see how they're stuck together in this one place right there. So that place is called the centromere, where the pair of chromosomes are stuck together, and that's why they look like little X's. Now metaphase is the next step. It's step number 2 in mitosis, and we're going to start with the cell we left off with that we got from prophase. You see the centrosomes. Let me change colors again here. The centrosomes right here are on opposite ends of the cell, and they have little finger-like projections coming out called microtubules. The chromatin has turned into chromosomes, so each pair of chromosomes looks like a little X because they're joined at the centromere. And during metaphase, what happens is you want to think middle for M for metaphase, because what happens is that while these microtubules are growing out and stretching across the cell, the chromosomes, they all line up in a single file, as you can see here, across like the equator of the cell or the middle of the cell. So the chromosomes all line up in single file in the middle of the cell in metaphase. So metaphase is M for middle. And here's more information about metaphase. Here's the microtubules. This is how you spell the microtubules, and those are the little fingers, and they're coming out of the centrosomes. And what they end up doing is they attach their free ends because they're coming out of the centrosome. So one end of the microtubule is coming out of the centrosome, but its free end ends up attaching itself right at where the pair of chromosomes stick together at the centromere. So on either side, you're going to have a microtubule sticking to that centromere area on one of the pair of chromosomes. And that goes for every single chromosome in there. Now, anaphase, we had prophase, metaphase, and now anaphase, so that's step number 3. So for A, think away, because we start with the cell that we just left off with in metaphase. See, all the chromosomes are lined up in the center, and these little fingers, these little microtubules have grown out and they've attached themselves to one of the pairs of chromosomes at the centromere region. So we're starting with this, the cell in this stage. And then what ends up happening is that these microtubules, these little fingers, since they're stuck to one of the chromosomes in each pair, they start pulling back towards their home, back towards the centrosome. So as you can see at the end result is these microtubules have pulled back towards their home, each in opposite directions, and they pull one of the chromosomes in each pair with them. So they take the chromosomes with them when they pull back to their home, to the centrosomes over here. So here's some more information about anaphase and a closer look at those chromosome pairs that are getting split up. Here's a chromosome right here. And you see, well, this is a chromosome pair. These are sister chromatids, and we went over that in the previous video. So this is a pair. So this is one, and this is another one. And they're called sister chromatids while they're still joined together. Well, if you notice, they form a little X because they're joined in the middle at this spot right here that I had purple. And that spot where they're joined is called a centromere. Now, around that centromere, around that spot, is like a candy wrapper of protein on each of the sister chromatids in the pair. So I call it a candy wrapper because it wraps around, it's protein and it wraps around one of the chromosomes' centromeres in the pair. And this protein is called the kinetochore. And so the kinetochore is actually where these microtubules attach to each sister chromatid, and it's these microtubules attaching to this protein, this kinetochore, is what enables them to pull them apart. And so you end up getting one chromosome on one side of the cell and one chromosome on the other side of the cell. So now these sister chromatids have been separated into each their own corners of the cell. And so now we don't refer to them as sister chromatids anymore, we just call them chromosomes. Now telophase, so we had prophase, metaphase, anaphase, and now we're in telophase or telophase. Some people pronounce it telophase, some people pronounce it telophase. So PMAT, and that'll help you remember the 4 phases. And T, the only thing I could think of that started with T to help you remember is the nucleus. Because remember at the beginning in prophase, that nuclear envelope, the walls of the nucleus, dissolved and broke down so the chromosomes could get out into the cell. Well, during telophase, the nuclear membrane forms again, but it forms around the chromosomes at each pole of the cell, on each side of the cell. So if you can, we start with where we left off with in anaphase. We have the sister chromosomes have been separated, so the pairs have been separated. One set goes to one side of the cell and the other set goes to the other side of the cell. But then if you look down here, now see the nuclear membrane or the nuclear envelope has reformed again and enclosed those chromosomes into the nucleus. So now the chromosomes aren't just loosey-goosey floating all over the cell anymore. They're contained within the nucleus. And that is the main point of telophase. At the same time telophase is going on, we have a subsection of telophase called cytokinesis. And if you notice this right here, this cell starts to cleave. They call it cleave, and you can also think of cleavage because well, it looks like cleavage, but that's the actual technical term is the cleavage of this cell because it puckers in like this, and then after a while, let's see, so it puckers in, and then it puckers in some more until finally they're two totally different cells, and this wall forms between them, so there's enough of a wall for them to both have a side, and then they're finally separate, two separate cells. So here it is again, cytokinesis. That is the subsection of telophase that cuts the cell in half, and that's just the act of the actual cleaving of the cell and cutting it in half. The part of the telophase, if you want to get technical, is the nuclear membrane forming around the chromosomes and containing them in the nucleus, making a nucleus again on either side of the cell. And that is the end of mitosis, which brings us back to what we went over in the very first video, the entire life cycle of the cell. Because see, here is where mitosis just happened. This little section of the cell's life right here. And so now it starts right here as a new divided cell, and it goes through interphase, and it grows, and it grows, and its chromatin gets replicated into sister chromatids, and then it grows some more before coming back to the mitosis stage where it will divide again. So here's just a reminder of the cycles of interphase. Interphase is three major parts. We have G1, which is the first growth phase after the cell is newly divided. The cell gets bigger because when one cell is divided in half, you're going to wind up with two smaller cells, right? So the G1 phase is so both cells can grow into big, full-size cells. And then the S phase, think sister, because that's the replication phase. So that's when the chromatin, the unwound chromosomes in this chromatin form, duplicates into twins, into twin sisters. So now you have two of everything. So those are sister chromatids. And then after the S phase, it goes into another growth phase. And the cell, including the sister chromatids, they all get bigger and thicker and get ready to divide once it goes all the way over and goes back to the mitosis phase. So that's it for mitosis. I hope that helped clear things up and gave you a couple of tips on how to remember all this stuff. It's really not that tricky. It's just a matter of remembering what happens first and then second and then third. And don't stress yourself out about it. So if you have any questions at all, leave them in the comments section or feel free to email me, and don't forget to like and subscribe. Thanks.